Okay, hello everyone. Thank you very much for joining us today for the BEEc Speaker Series for February. The BEEc Speaker Series is our international webinar that is organized by BEEc. So for those who may not know, BEEc is the Center for Bee Ecology, Evolution, and Conservation at York University. We are an organized research unit. If you'd like to learn more about us, you can visit our website, www.yorku.ca/bees, or you can email me, the BC coordinator, at bc@yorku.ca. We are an initiative that strives to advance research in the fields of bee ecology, evolution, and conservation. The mission of BEEc is to foster interdisciplinary, innovative, collaborative, and cutting-edge research in order to advance our knowledge on bees and implement policy changes to help sustain pollinators globally. Our goal is to apply our collaborative efforts to the development of policies and environmental management for the long-term sustainability of bees and the bioecosystem services they provide. If you would like to hear more about BEEc, feel free to join our listserv by sending an email to listserv@yorku.ca with the command in the body of the email stating subscribe bees and then your first name and last name. You will be added to the list. You will be notified of any upcoming programming, receive a monthly newsletter, as well as reminders of events such as the BEEc Speaker Series like the one today. If you are a York University researcher or collaborator, feel free to contact me for more information about applying to be an associate. Our associates are interdisciplinary researchers and community members that are involved with pushing the limits in bee research, education, public outreach, and policy. And you can see here a small selection of our BEEc associates. So today you have joined our BEEc Speaker Series program. We run this program once a month, typically the second or third Friday of the month at 11 a.m. EST using Zoom. We've already had a great selection of speakers starting in the fall, and many of these talks are available for viewing on our website as well. Feel free to visit www.yorku.ca/bees/speaker-series to learn more about this program and also to register for any upcoming talks in the following months. So just a few logistics for today. We are using the Zoom platform and only panelists can turn their camera and microphones on. We ask that you please use the Q&A box to pose questions to the speaker, and we will pose the questions back to them at the end. Please use the chat box sparingly for general comments as it can be distracting for the speaker to continuously having notifications popping up. If you're experiencing any issues, you can send a private message to myself or email to bec@yorku.ca. If you need to leave at the hour mark and we are still in the Q&A discussion period, do not worry because this presentation and the Q&A session will be recorded and posted at a later date after the contents of the talk are published. The talks are also posted on our YouTube channel at youtube.com/@beesyork. We have several previous talks already available on the YouTube channel to view. We also have talks posted from other programs like the BEC Associate Series, as well as BEEcon, our annual conference. We invite you to subscribe to our channel so you'll be notified when our next video is posted. And before we get started today, I would like to recognize that many Indigenous nations have long-standing relationships with the territories upon which York University campuses are located and precede the establishment of York University. York University acknowledges its presence on the traditional territory of many Indigenous nations. The area known as Takaranto has been caretaken by the Anishinaabe Nation, the Haudenosaunee Confederacy, the Huron-Wendat, and the Métis. It is now home to many Indigenous peoples. We acknowledge the current treaty holders, the Mississaugas of the Credit First Nation. This territory is subject of the Dish with One Spoon Wampum Belt Covenant, an agreement to peaceably share and care for the Great Lakes region. If you would like to know more about the history of land that you come from or where you are joining us from today, this website is a fantastic resource for this exploration. It was a Canadian initiative, but now has a worldwide coverage. And now I would like to welcome our guest speaker, Dr. Nadia Tsvetkov. So Nadia is a postdoctoral research fellow at the University of British Columbia in Dr. Leonard Foster's lab. She completed her PhD and her master's degrees both with distinction at York University under the supervision of Dr. Amro Zaid. She has received an NSERC PDF award and she published in prestigious journals, including the journal Science. Her research on honeybee health and behavior received wide media attention and impacted government policy. And with that, Nadia, if you can please share your screen and take it away.
Thank you very much. Excellent. Okay. So today I'll be talking about quantitative behavioral analysis, and I'll be showcasing some of my own work, some of other people's work, and I also want to talk about kind of the field in general, where we are and kind of where we're heading. So one of the beauties of quantitative behavioral analysis is we take complex phenotype and we reduce them to measurable responses, which enable us to study them systematically. So a complicated phenotype like learning becomes as something as simple as whether or not a honeybee extended her proboscis. Also, I want to note that most of the work that I've done on honeybees, so I will be speaking mainly about honeybees, but a lot of these principles are generalizable, of course, to other bees and insects and animals in general. And this is really neat because measurable responses are really, really useful. So for example, when we measure proboscis extension response in honeybees, we can also measure something called sugar responsiveness. So we take a sugar solution, we dap the honeybee on her antenna, and we see whether or not she extends her proboscis. And we can do it with multiple different sugar solutions and see to which ones she responds, right? The ones that contain more sugar or less sugar. And we know, for example, in this graph, you can see a really neat correlation between sugar responsiveness and how well a honeybee learned. Now, when I say in this instance, a honeybee learned, that means a type of classical conditioning response. So we harness the honeybee, we give her a puff of odor, we touch her antenna with the sugar solution, she extends her proboscis, we let her feed. And after a few of these pairings, whenever we give her the puff of odor, she automatically extends her proboscis, even without us touching her antennae. And here we say she has learned to associate the odor with the sugar reward. And using this relatively simple paradigm, we can actually measure a bunch of different types of learning. So acquisition, latent inhibition, extinction, by kind of varying the trials and the order we do things in. So really, really, really useful. And of course, we can do all wonderful statistics on it and investigate things that would be really hard to do otherwise. So for example, in this study, what I did is I took a bunch of bees from emergence, and then I either placed them in isolation, a group of 8 or a group of 32, let them grow up for 7 days, and then I measured their sugar responsiveness and how well they learned. And I found that isolated bees had worse learning and memory than bees raised in groups, but also they had higher sugar responsiveness. Because I was able to do this on dozens of individuals in this paradigm, we also took the brains of these honeybees and looked at the neurotransmitters in them. And we found that the group size that the bees were raised in had lower dopamine levels. Now, this is really neat because we actually have quite a bit of literature that shows a direct effect of lower dopamine that increases sugar sensitivity, and lower dopamine, which decreases learning, has no effect on memory, which is exactly what I found in this study. What's really cool, it's also the same thing we see when we isolate mice and rats, which might mean that there's some kind of conserved evolutionary kind of thing to social groups, the neurotransmitters, and then to the phenotypes that we observe. Another cool thing that we get from this kind of relatively simple, straightforward experimental design is that there's evidence that social evolution involves in changes in regulation of the reward system. So sugar sensitivity is one of the things that allows us to measure the reward system inside of honeybee. This is some other papers I won't go into right now. But it looks like, that modulation of the group size also modulates the reward system. And kind of another paper experiment, I really want to do this, is repeat this experiment with facultatively social bees. So bees that are sometimes solitary, sometimes they're not. And then with solitary bees and see whether or not their sugar responsiveness changes and whether or not their learning is affected and what does dopamine is doing in their brain. I have a theory, I have a hypothesis rather, that it's not that isolation is detrimental, but that group is beneficial. And there is a distinction there, especially when it comes to social evolution. Again, but this is a hypothesis to be tested and kind of the methodology to be worked out. But even though we have this kind of, we take these complicated phenotypes and we reduce them to these kind of neat, easily measurable responses, we have to remain vigilant and careful. So from other studies, and as well as my own, so in my own study, you can see there's a correlation between sugar responsiveness and learning. Sorry. Other studies have shown that it's actually a direct effect. So if you mess with sugar responsiveness, you'll get bees that learn worse. And there were quite a few studies that investigated whether neonicotinoids, which are a commonly used pesticide, affect learning in honeybees. But a lot of them didn't measure sugar responsiveness. So without measuring sugar responsiveness, you're unable to tell whether neonicotinoids affected sugar responsiveness, which is a perception issue, or learning, which is a processing issue. Certain studies actually did measure sugar responsiveness, found an effect. So neonicotinoids reduced sugar responsiveness, but didn't account for it in the model for their learning behavior. So you couldn't tell which one is it because the stats weren't done properly. So at that point, we actually didn't know whether neonicotinoids affected sugar responsiveness or learning. So we really have to be careful when we take these measured responses that we're actually measuring what we think we're measuring. So this is where I came in, and I did this experiment where I exposed honeybees to clothianidin, and indeed, clothianidin bees had lower sugar responsiveness than the control bees. But when I did the learning part, so A is pairing sugar reward with a 1 cent, and panel B is pairing a different type of odor with salt, which tastes bitter. So the honeybee has to learn the difference of when to extend her proboscis and when not to extend her proboscis. And when I did the stats, I found there's actually no treatment effect on learning, but there is a statistically significant effect of sugar responsiveness, which is to be expected. Now, might seem a bit trivial, but it's really critical, especially when you're trying to figure out how neonicotinoids affect the bee. What are the molecular mechanisms behind these? And one of these things is I also did proteomic analysis on the bee brains that I analyzed, and I found downregulation in treated bees for biosynthesis of amino acid and carbon metabolism, and upregulated proteins for RNA translation and ribosome. But more importantly, I was able to identify nicotinic acetylcholine receptor alpha 7, which is one of the components that makes the receptor that neonicotinoids bind to. And this receptor subunit expression levels had a very nice correlation in clothianidin-treated bees with sugar responsiveness. And as you can see by the row there, that's a pretty good row. That's a very tight row, actually, even though the spread around the line seems like it's a lot. And what does that mean? Well, nicotinic acetylcholine receptor alpha 7 subunit is expressed in the antennal lobes of the brain, which are the lobes that receive input from the antennae. They are not expressed in the mushroom bodies, which are kind of the cognitive processing of the brain. So this is another kind of line of evidence to suggest that neonicotinoids are really messing with perception rather than processing of the cognitive process. And this is critical because um, sugar responsiveness has other effects besides learning. So highly sugar responsiveness, bees in sugar responsiveness, they also tend to collect more pollen. They tend to collect more water. And they tend to be also responsive to other kind of stimuli. So we really have to be careful that we're measuring what we think we're measuring. So this is, again, interpreting the results, we have to be careful. So if we want to measure phenotype A, we choose a measurable response. And then when we make back our kind of interpretation of results, we have to be careful with when we go back to phenotype A, that it is actually it that we measured. Sorry, so with a clearer example, it's we want to measure learning, so we use proboscis extension, but then turns out that we might have not have measured learning, we might have measured sugar responsiveness. So we have to make sure we design our experiments carefully and we take into account all of these things. Another part of the problems of result interpretation that I see with quantitative behavioral analysis is when we take the measured response and we say it represents the entire phenotype. So here we tend to do this in abstracts and especially titles. And this creates apparent contradictions in the literature. So for example, you can go into the neonicotinoid literature again, which is pesticide, and find that neonicotinoids do in fact affect the immune response of the honeybees. And then you'll find other papers that say they don't. But if you actually look at what those papers measured, you will find that, well, one paper looked at viral replication, while another paper looked at gene expression of the immune genes. And they're not actually contradictory to each other because an immune response is a huge, complicated phenotype. So they might be different in this particular response to this particular dosage in this particular duration. So we have to be careful about this, not just because it creates confusion in the academic world, but because, of course, then, pesticide companies go and say, well, look at this. The academic literature is a mess. They don't know what they're doing. They have contradictory results all the time. So I think we have to be careful where we are clear, precise in our language when we write these papers. Now, another thing about kind of quantitative behavioral analysis is it's nice that we can measure simple responses, but how realistic is it? So, for example, obviously the harnessing of the honeybee is really not something a bee will experience naturally. But also when we look at foraging, we tend to look at feeder visitations, but a feeder often lacks a lot of the signals that a flower patch will have, right? The scent, the complicated arrangement of other flowers nearby, and things like that. So another way of doing kind of more realistic scenarios is just by simply observing the bees. So the old-fashioned way is you get a few undergraduate students, you get an observational hive, and you get them to scan the hive every few minutes and record what the bees are doing. You can mark your different treatments or group of interests with different colors so they can tell, a green bee was waggle dancing at this time for, this long. This is pretty neat. This is a very, very useful technique that I've used before. But of course, it has issues. So some of the limitations is that actually your undergraduate students can produce variation in the data. And I have observed this where you get all of this information, and then it turns out that the student is a statistically significant variable in explaining some of the data. So this is a problem. Of course, you're usually limited to working hours. You're not gonna make your student to come in at 5 a.m. to see if the bees are dancing. You need daylight, or at least some sort of light. Now, normally a honeybee colony is dark inside, it is closed, so light exposure might change their behavior. And you have limited individual differentiation. So you can color your bees basically on day of emergence or on treatment group, but you're usually limited to a few dozen colors. And this is where technological advancements really comes in. So here it allows you to measure hundreds of individuals continuously over time. You don't have to expose the colony, the insides of the colony to light. And of course, you don't have observer effect variations. So this is an experiment I did. I used RFID chips. So I glued little RFID chips to honeybees. I placed a RFID sensor on the colony, and then whenever a bee passed through the sensor, I recorded it, and then when she passed back inside the colony, I recorded it again. So I was able to measure time outside the hive, which is effectively their foraging time. And here you get this kind of medium-sized data where from my recording, I got about 100,000 rows. After data cleanup, I get about 7,000 individual flight times for different bees. I forgot to mention that each RFID chip has a unique identifier that the reader can read, so I get individual results for each bee. Now, for this particular experiment, I had two treatment groups, one control and one treated bee that was treated group that was treated with clothianidin, a neonicotinoid. And I was interested in how foraging flights might differ. And here I used a generalized linear mixed model where I found that treated bees, as they aged, took longer flights than control bees. You can see also that the treated bee line is shorter, and that's because their lifespan was shorter. You can see that I'm plotted, so this is about the 7,000 data points that I had. And of course, here the statistics you use is critical. Now, since I was just looking at two different treatment groups, I used a generalized linear model. But I could have also used a generalized mixed model, additive mixed model. So a generalized additive model is a generalized linear model that doesn't assume linearity. And when I apply that to my data, here's how it looks. So I still have this difference between treatment and control. But you can see that especially in the control bees, the flight time pattern is very different than the assumed flight that I see in the literature a lot, that bees just fly longer and longer as they age until they don't return to the hive. Turns out that bees fly longer and longer until they don't, until they actually start taking shorter and shorter trips, and then it kind of evens out. This is really critical, especially when we study life history traits. For example, on one side here, you see the paper by Dukas, who used the old-fashioned way of marking bees and tracking them to find out what's the mortality rate of foragers. And he found that by about day 18 of foraging, most honeybees died. But if I look at my RFID data, I have some foragers that lived past day 50, which is really long, kind of at least what we assume is really long, because most of the literature you find, it says, well, once a honeybee starts foraging, she's alive for about 2 weeks. Um, and here I have several bees that are alive and well in the control group at 40 and 50 days after starting foraging. So these continuous long-term observations really enable us to have more accurate data. Okay, so now with another kind of advancement in technology, we get this behavioral work and this big data um, quantifiable analysis. So here we individually barcode honeybees. We can barcode, 1 to 2,000 bees simultaneously. In this particular paper, they took 1 frame per second for 7 days. So you can imagine that's a lot of pictures. That is truly kind of big data now. And you have automatic detection of behavior. So in this case, it was an algorithm, and they were able, in panel B here, in the purple square, you can see that it identified trophallaxis. And this is really cool because trophallaxis happens all the time in honeybee colonies. It's critically important for signaling, but also transferring of microbiomes and immune information. And it's actually really hard to study because you kind of have to observe, if you're doing this by eye, you have to observe thousands of bees simultaneously. Another really cool paper published, they did 1 month of observations and got 27 million images of the queen and her surroundings. And the way they did this is they actually built 2 robotic arms. So right here at the bottom, you can see it's 2 robotic arms with 2 cameras attached to each arm on each side of the observational hive. And this camera would find the queen, track her, identify her, identify the court and what they were doing, and then look at the contents of the frame and as it changed over time. Really, really kind of cool observations. Now I also want to talk what we do with this kind of technology. So one of the earliest papers that we have, it might be the first paper that we have with this kind of tracking, is this paper that, again, barcoded the bees, had automatic tracking, and then looked at the waggle dances of the bees, and the algorithm was able to decode it, and right here on the image, plot where the honeybees were pointing to which feeder, right? So the honeybees have this waggle dance where in the dance, the forager comes back and she tells other bees of where the food source was, how good the food source was, and kind of how hard it was to get to, right? So kind of both the distance and the angle where they need to fly to reach this food source. So if you're doing manual observations of the waggle dance, it's really hard to do because getting the correct angle relative to the sun at any given point is difficult using kind of eyeballing it. But with automatic systems, with algorithms, you can really get it precise. And they were able to map this really cool thing where they were able to pinpoint where the bees were flying. Now, it's been 10 years since this paper is published. Since then, it was cited by about 60 papers that were doing something with this type of technology. So it actually has more citations, but some of those other citations just did kind of the old-fashioned types of experiment and just cited this paper either in this discussion session or something else. So 14 of those 60 citations are actually by reviews or perspectives. And I don't want to be too harsh here because I think this is kind of common for newly developed technologies or methods. So for example, conservation genomics, right? It's when we use genomics and transcriptomics and apply it to conservation in field questions. And for a long time, we had perspectives and reviews that said, yes, we can do it, we should do it. Here's a whole bunch of advantages of this method. But there were no papers until kind of we got together with some people at York, and we were able to apply this conservation genomics techniques to a declining bumblebee. So I don't want to be too harsh. This is kind of normal. Another 35 of those 60 citations were technology development. So either the same lab or other labs that made up their own tracking, used other barcodes types, other types of algorithms, and so on and so forth. Now, again, this might be normal when a new method and a new technology really takes off, but more than half of the citation are just technology development. And when I say just, I don't mean it's not a lot of work. What I mean is the paper is only is there to showcase the technology and prove that it works. But then what do you do with this? And there were only 6 papers that actually did some sort of hypothesis testing. And again, this has been around for 10 years. This seems woefully a small number. Now, some of those papers are really cool, and I want to showcase them because I think some of those papers really take advantage of the benefits of this technology. So one such paper looked at how the worker bees behave when there's no queen around. So when a honeybee queen is around, she's normally around in the honeybee colony. Mainly only she lays eggs. If you remove the queen, the worker could start developing egg laying, um, egg laying, yeah. So they took away the queen and tagged the bees, and then tracked whether the honeybees were laying eggs, and then which ones were going out to forage. And they found that some bees were exclusively egg laying, some bees were exclusively foraging, and some bees were doing both. Some bees were foraging and then egg laying when it wasn't peak foraging time. And they took this information and they used it to look at differential gene expression and differential chromatin accessibility. I won't go into the differential expression part, although there's some really cool stuff. So if you want to read the paper, please do. Here, what I wanted to point out is that this was a really good use of this technology because it's continuously monitoring the bees. And if you didn't have this continuous monitoring, you could have easily mistaken a general bee for either an egg layer or a forager, right? Because you can see that some of the generalist bees lay eggs very kind of biased towards certain times. So if you're using, collecting bees at 6 a.m., you might assume that a generalist bee is actually egg laying bee. So precise continuous monitoring allows you for precision in categorizing your bees. Another cool paper actually has to do with drones. And I have to admit that part of the reasons why I think it's cool is because drones are a notoriously understudied cast in the honeybee system. We most of the time just ignore their existence. And here in this paper, they tracked how fast the drones were moving. So on the colored panel, the brighter green colors are when the drone started to move fast. And on the black and white panel is the black lines is when the drones left the hive. So honeybee drones, they don't forage. They only fly out when it's time to either practice their mating flight or they're actually going out there for a mating flight to try and find the queen. And they found, by analyzing this data, they found that these clusters of activity before the drones are ready to leave the hive. But also they found that it's both an internal cue and a social cue. So drones use 2 cues to figure out, is it time to start flying? Of course, there's other things that affect them. For example, weather, right? If it's bad weather, they don't go out flying, they don't become hyperactive. Okay, but as cool as these things are, there are problems with these methods as well. So part of the problems is, of course, the cost of setup. So you have the cameras, if you want to do robotics, the AI or algorithm development, and of course the storage. Storing 27 million high-resolution images is not trivial. There's, of course, the cost of processing. So you need servers or you need cloud computing, and it might take you weeks to process this amount of images. You need kind of very particular skill set of bioinformatics and maybe AI image recognition um, to actually start analyzing these results. The other problem that I see in some of the papers is sometimes they put the cart before the horse. So in quantitative behavioral analysis, we need to figure like kind of the step one is which phenotype we want to look at, and then we kind of try and reduce it to a measurable response. Here it seems like we get a measurable response, but it seems to be disconnected from like any biologically relative phenotype. So for example, in this paper where they tracked the queen, in panel G, the purple bars represent where the queen was orienting when she was resting and not walking. And you can see that most of the time she was kind of, her head was up, she was orienting up. Okay, but what does this mean? Right? Like, okay, I see that the queen most of the time gets her head up when she is not walking. How is this biologically relevant? So I really think that the technology, we need to use the technology and actually go back to hypothesis-driven experimental design and go back to this idea of complex phenotype and then go back that we're interested in and then look for the best technology, best tools, best methodologies to measure the response. And not the other way around is like, here's the cool piece of tech I have. Let's see what we can do with this. So one of the things I want to look into is risk assessment and how honeybees assess different types of risks and how they deal with individuals who are low or high risk. So the complex phenotype is risk assessment. The measurable responses I want to measure is things like waggle dance, feeder visits, and recruiting success. So on previous studies, we see that risk assessment in honeybees is definitely present. So for example, when we get a predator like a wasp near a feeder, bees are less likely to return to this feeder. And some foragers will use a stop signal, to interrupt a waggle dance by a different forager when they encounter a predator, right? So a forager will be dancing and this bee will tell it to stop. However, despite all of this, there's some foragers who persist going back to this high-risk feeder. And I want to look at these high-risk versus low-risk individuals. So, for example, I want to ask, do high-risk dancers adjust their dance when predators are present? So my hypothesis is even in these high-risk individuals, the dance after they encounter a predator will indicate that it's not such a great reward than that individual's dance before they encounter a predator. So I do think they will adjust their quality of the reward based on predation risk, but they'll still think it's good enough to risk it and go out there and try. The other thing I want to look at is what do low-risk foragers do when they use a stop signal, but the high-risk dancers keep dancing. So one of the things that could possibly happen, one of the hypotheses I have, is that these low-risk forager might revisit the site, assuming that it's safe, right? Because if you're thinking if somebody is like dancing and tells you, obviously humanizing them here a bit, that like, oh no, this place is great. And you go and you're like, no, stop dancing. This is really, really risky. There's a predator there. But this individual keeps dancing. Maybe the low risk bee will assume that, oh, maybe the predator has moved on. Maybe this bee has information I don't have. And another thing I want to do is this high risk versus low risk genomic and transcriptomic analysis. And this will allow me to look into which pathways, molecular and genetic, are responsible for risk-taking behavior. Now, this might seem like a grandiose plan, but in honeybees, it's actually very much doable, especially within a single generation. So this is something I've done already for tolerance to neonicotinoids. So I exposed a bunch of bees to a neonicotinoids and then seen how many of them died and how many of them lived. And then I genotyped them individually to figure out who the father is. Because honeybees are polyandrous, and in a single colony, you have one mom, but you have like 20 or 40 different dads. And when I did this for this tolerance and clothianidin, I found that yes, patrolline definitely has a statistically significant effect. And we can actually calculate broad sense heritability. And we found that you have a pretty significant heritability for survivorship. Then we took another set of bees and we exposed them to a low dose of clothianidin, one where they didn't die, but they were from the same batch of bees. So then we also patrolined those, and we were able to tell which of those bees come from which patroline. And we found that between patrolines that are whose workers are likely to die and those who are not likely to die, there is differential gene expression. And most of those gene expression is a difference in their gut. So we in the Malpighian tubes specifically, actually, which are the detoxification kind of the equivalent of kidneys in humans. And in those Malpighian tubes, we saw signals of degradation and apostosis in the bees that came from high mortality petrolines. And then we took those bees and we actually sequenced detoxification genes, and we found non-synonymous mutations in certain haplotypes that strongly correlated with survivorship. And as you can see here, haplotype E, which is at the bottom, it actually had a deletion, which basically the read codon basically moved, so it became kind of nonsense, and then it reached a stop. So it had a truncated detoxification gene, and that petroline with this haplotype had the lowest survivorship rate. And while this is we can do this in a single generation in honeybees, which is really cool because a lot of other systems, you need multiple generations to figure out heritability. This particular paper, I tested 863 bees made it to the paper. I tested probably over 1,000 bees because, of course, not all of the bees ate all of the sugar solution with the clothianidin in it, and then you have some other parameters for quality control. So, of course, not all the bees make it. Now, for something as kind of a simple time point of whether or not a bee survives, so that's quite a clear phenotype. I was able to test 1,000 bees in the summer. I cannot test 1,000 bees for even the kind of extension response learning paradigm. Even if I had 10 undergrad students, I don't think I have physically the space in a lab to do that. And definitely not as something as complicated as risk-taking. I would need like at least 1,000 individuals to do this, and doing it by hand is impossible. But this is where I lean into the benefits of these tracking systems. So you track thousands of individuals, you track them continuously, which allow you to categorize them accurately into high and low risk individuals. And you can track multiple behaviors at once. So I can track the waggle dancers, I can track the bees that tell them to stop, and I can track the bees who are recruited by the waggle dancers and then see what they do. So hopefully I've convinced you that we have a lot of kind of new technologies and different methods to do quantitative behavioral analysis, but we have to be careful not to fall into, oh, I have this technology, let me do something with it, rather than go back to strict experimental design of here's my hypothesis testing, how I'm gonna test it, which method is the best, and actually pick the best method for this question. So thank you all for listening. Thank you for UBC and York, where I've done a lot of this work, my funding agencies, NSERC, OMAFRA, OGS, Project APSM. Thank you for all the Bs that enabled my research. And again, thank you for listening.
Thank you, Nadia. Great talk. So we'll leave it open for anyone to have questions. You can post the questions in the Q&A box. In the meantime, I have a question. So you
Yep.
were mentioning how there's that statistical variation when you have different students doing the traditional sampling techniques. Now, with the onset of all these new technological advancements, everyone's kind of using, I'm assuming, different algorithms and a different type of microchip or a different recording or a different frequency. So how much of that do you think is also going to show a statistical variation in just how the recordings and the data interpretation is done?
Yeah, that's a great question. So probably quite a bit. Now, the saving grace is that if you're comparing, two different groups of bees, you're using the same system to monitor both of those groups because you're doing it in a single experiment in a single lab. But yes, the type of algorithms we use is really, really important. So, for example, if we encountered this in proteomics, there was just a paper published on it where one version of the software on Windows gives you different results than that software on Linux. And when it was brought up to the writers of the software, they said, well, we had to change the math because this type of math doesn't work on Linux when it works on Windows. So different math equals different results. Now, this is kind of a little scary when you're doing protein identification because, well, you're hoping to actually get a clear picture of what's inside a bee. And here you don't know which one is more accurate, the Windows or the Linux one. You don't know.
No, so that's just something everyone will have to sort of work, or maybe even, I guess, is it feasible? Maybe not feasible to even use multiple technologies because you'd have to use different chips at the same time. And I don't think that would work.
Yeah, it's hard. I think eventually maybe there'd be a study where you can compare the different algorithms to each other.
Hmm.
But they have different requirements for the barcodes. They have different requirements for the lighting, for how much you can see the bee against the background. Yeah, it's part of the problem of this technology is there's a lot of variation and it's going to be very different lab to lab. And I think it's going to happen for a few more years until the technology advances where you won't need kind of these strict parameters for the AI or the algorithm to read the barcodes and identify um, the behavior.
Hmm.
Now, again, the cool thing is that it doesn't matter that much if you're doing a single experiment within comparing two treatment groups. And I mean, we know this for even for other more established technologies like qPCR, you might have differences between students, between labs, in the extraction methods, because you can still use different chemicals to extract different procedures. You might get some variation between labs in there as well.
Right. Any more questions?
Hey Nadia, I have one.
Yeah.
How would this translate? I guess your work's been largely done in honeybee, maybe a little bit in bumblebee. How would this translate to other bees? Is this technology amendable to solitary bees or facultatively social ones? And how could we branch
Yeah.
out?
Yeah. So that's a good question. So part of the advantage of kind of more solitary bees is you don't actually need to track thousands of individuals. So that makes the labeling and the algorithm to detect them easier. So you could actually get away with marking them in simpler ways. The problem you get is depending on the nest type, right? Because if you want to use imaging detecting, you'll have to physically see into the cell. So depends on how well you can make it. But I actually would assume that it's probably easier in solitary bees. So if you have your solitary bee in a twig and you kind of put some plexiglass on the side and you can record her continuously, she has less problems with crawling over other bees and then the algorithm can't see what the bee is doing, going on the other side of the frame so the algorithm can see what the bee is doing. um, and probably the type of things she engages in is simpler. So the algorithm has to choose, like you code it, obviously. Is she cleaning? Is she feeding? Is she resting? Right?
Mhm.
So uh, it's probably easier to implement for solitary bees, assuming you can get access to the nest. And again, if it's a solitary bee, you don't actually have to mark her. You just know this is the one individual, and that's it.
Well, yeah, but I guess yeah, the marking because the bees get smaller, right? The
Yeah,
like afflicted and a lot of the aphid bees are small, and so they're like any kind of individual tracking could be very difficult so far. I think the technology hasn't gotten small enough for that, is my understanding. Is that correct?
no, that's correct. Yeah. But if you have like only a few bees, so like three, five, you can mark them with just paint. And even in infrared, which is how these pictures are taken, so it's kind of dark,
Mhm.
you can find paint that is different in infrared. And
Mhm.
so you only need, four or five different paint markers on each bee. It doesn't have to be large, it just has to be visible in order to tell those bees apart.
But overall, the technology still requires that kind of 2D.
Yeah,
So the honeybees are amenable to that. You can make an observation.
that's
Hi,
right. So
bumblebee.
part of
Yeah,
the bumblebee, part of the problems with there are a few technology developments for bumblebee, but part of the problem is they tend to build their nest in kind of this clump ball.
yeah.
So yeah, so you either have to have multiple cameras, which introduces quite a bit of issue with image reconciliation. And of
Yeah.
course, kind of like you can't see all the bees all the time. Yeah. But I mean, the people who developed this technology were honeybee researchers. So they, of
Yeah.
course, kind of used it for honeybee research.
Yeah, I think there's application, but yeah, it just gets a little bit more. I think as
Finicky, but
technology is improving, we're getting better and better, smaller tracking. So if we could just individually monitor with like probes, even we could like maybe do away with strict video sensing. It could be more like, I don't know. I think the
yeah,
technology does kind of expand beyond the human brain at some point. So yeah, there should be some.
yes, and I actually think that like for solitary bees or like with when there's very few bees in a nest, I actually think it's easier because part of the problem is the number of bees and they're constantly moving around. That's part of the problem the algorithm has with.
Mhm.
But if you actually simplify it to a few bees, I think it's actually going to be a lot simpler.
Mhm. Yeah, well, we could talk about this offline
Yeah,
too.
yeah.
Yeah, great talk. Thank you.
Thank you.
Was there any other questions in the chat? No, not today.
No.
Quiet, lots of people. I guess they're having their lunch. But yeah, thanks so much, Nadia.
Thank you.
I'll just quickly remind everyone for next month, we have another speaker, sorry, Dr. Monica Egger on March 21st at 11 a.m. So if you haven't already, you can visit the website to register for that talk. And with that, I just want to thank everyone for joining today's seminar and see you next month, hopefully. Thank you.